In this video lesson, we're going to continue our discussion on proteins by focusing on a specific type of protein called enzymes. We'll also spend some time at the latter half of the video talking about some of the techniques we use to study and explore proteins. So proteins have a lot of different roles. They can provide structure. They can act as enzymes, so biological catalysts. They are hormones. Hormones can be proteins, and there's many other things. So this diagram highlights some of them. So again, the surface of our skin is made of protein, so it can perform structural. Our hair is made of protein. Insulin is a hormone. Amylase is an enzyme. We have the pigment that makes up our blood cells that gives our blood the red color, so hemoglobin is a protein. Bones have minerals and they're embedded in collagen, which is a protein. Muscle fibers are protein, tendons are proteins, toenails and fingernails are made of protein as well. So there's two different shapes of proteins or groups of proteins that help determine their function. So there's fibrous proteins and there's globular proteins. Globular proteins have more of a spherical shape. They're globy. That's how I remember them. They are soluble because their R groups are hydrophilic on their outer regions, and then they keep the hydrophobic ones towards the center, as you can see in the yellow and the blue. These proteins tend to act as hormones, enzymes, and transport molecules. And they are sensitive to high temperatures because when the temperature rises, some of their intermolecular forces will break. So the London forces and the hydrogen bonds will be broken and then they'll unravel. So high temperatures are not good for these types of proteins. Fibrous proteins, on the other hand, are long linear bundles of polypeptides, and they'll be held together by either covalent bonds or disulfide bridges. So they're almost roped or threaded together. So in this case, there's three polypeptides coiled together and they form a triple helix, and then covalent bonds are being held between the polypeptide chains, and there's lots of hydrogen bonds holding them together. These types of proteins aren't soluble in water because of their tight packing together. So you think about an example of a fibrous protein, our skin and our hair. It's helpful that they're not soluble in water because we don't want them to dissolve away. So these proteins provide strength and structure. So they are strong and they have a structural role. They're also less susceptible to heat. Again, we can expose our hair to heat when we straighten it and so on, and it doesn't damage it permanently. So enzymes. Enzymes, if you remember from biology, are biological catalysts. And if we remember about what catalysts provide an alternate pathway that has a lower activation energy than the uncatalyzed reaction. So one common misconception is that we say that it lowers the activation energy of a reaction. No, it provides another pathway for the reaction that will have a lower activation energy. So they're known as homogeneous catalysts. So they are homogeneous catalysts. They're in the same phase as the substrate, so these reactions will happen in aqueous solution. So we're not going to take a long time writing about the lock and key and induced fit models because we learned about them in biology. But if you remember, there's two different models that we use to explain how enzymes work. The first one was the lock and key model. So we say the enzyme is the lock, the substrate is the key, and the active site is where the key fits into the lock. So in this example, there's a spot for the substrate to bind. When they bind together, that's the lock fitting into the key fitting into the lock, and that forms the enzyme substrate complex, and then the reaction occurs and then the products are released. So that's the lock and key. And this explains the specificity for an enzyme for its substrate because only certain keys can fit in the lock and be unlocked. The induced fit model, on the other hand, is similar, but it's more like once the enzyme starts to bond to the substrate, the enzyme will now close or mold around the substrate. So they aren't an exact fit until they've join together. So the substrate induces the fit of the enzyme to it. Again, in biology, we learned about enzyme inhibition, and there's two different forms. So in competitive enzyme inhibition, the inhibitor blocks the active site. So now the substrate can't bind because the spot is blocked. And then non-competitive inhibition is when there's an inhibitor that binds at an allosteric site, a site that's not the active site. And then that changes the shape. And then the substrate still can't bind because the shape of the active site is hindered. So both of these types of inhibition prevent the substrate from binding and prevent the products from being produced. So if we needed to calculate the rate of an enzyme reaction, the initial rate is dependent on both the concentration of the enzyme and the substrate, so it's bimolecular. But the concentration of the enzyme never changes, so meaning that it never gets used up. So the concentration of the substrate is the thing that has the more impact on the rate. So the rate is equal to K times the concentration of the catalyst times the concentration of the substrate. So if we look at Michaelis-Menten kinetics, when you're increasing the substrate concentration, the rate of reaction will increase, and it'll increase quite steadily until there's no more enzyme that's available to bind. So eventually, increasing the substrate concentration will have no further impact on the rate of reaction. So that's known as the Vmax. This is the halfway point, and the concentration of the substrate is being shown that it's increasing. So the Michaelis constant, or the Km, that's the concentration of substrate for half the maximum rate of the reaction. So you find that on the graph. And if that number or value is low, like this one, there's a strong affinity to bind to the enzyme. So it takes a very low enzyme or substrate concentration to reach the maximum rate. And then if that value is higher, like for enzyme number B, there's a lower affinity for the enzyme, so it will take a high concentration of the substrate to reach the maximum rate. So you can see the differences here. So you don't need a lot of substrate to get to the maximum rate, whereas for the second one that had a higher Michaelis constant, you actually need more of the substrate to get to the maximum rate. So competitive inhibitors, you can see that's the red line here. It doesn't change the maximum rate of reaction, so you eventually can reach that maximum rate. But what it does do is change the Michaelis constant. So it changes the shape of the reaction. So instead of this steady curve like so, it levels off and takes a while to get to the maximum rate. With non-competitive inhibitors, if you had to point that out in the on a graph, this is the no inhibitor again. This actually will impact both the Michaelis constant and the maximum rate of reaction. So a non-competitive inhibitor changes the shape of the enzyme, so it doesn't matter how much substrate you add, the shape of the enzyme has altered. So now you can't reach the maximum rate of reaction ever until that inhibitor is removed. So this table summarizes the two differences. So what I've just talked about in the last two slides, competitive inhibitors bind at the active site, doesn't affect the maximum rate of reaction, but it does affect the Michaelis constant. Non-competitive inhibitors bind at the allosteric site. It decreases the rate of reaction, the maximum that you can reach, but it actually doesn't affect the Km. So we'll back up because I didn't talk about that before. You can see that the half max is still at the same spot on the graph. It just impacts the maximum rate. So it never can get to the maximum rate, but the Michaelis constant stays the same. Again, in biology, we learned about end product inhibition. So remember, metabolic pathways are multiple reactions and they're catalyzed by multiple enzymes. So sometimes the end product in a metabolic pathway will actually act as an inhibitor on the initial enzyme. So we started with our initial substrate and then we made an intermediate. There was another enzyme. We had a second intermediate. Then we get our end product. So 3 enzymes catalyze this reaction. Now that we have our end product, we don't want to waste energy to keep making it once we have it. So that will act as an allosteric inhibitor or a non-competitive inhibitor sometimes, and will inhibit the initial. And then we won't continue making the product because we already have it. So one of the requirements for your option is to identify ways that we can study polypeptides. So there are 2 methods that we can use. We can use chromatography, which we learned about when we were studying pigments in our photosynthesis unit. And we can also use gel electrophoresis, which we studied in biology as well when we were looking about sequencing or exploring DNA profiling. So we can study the composition of proteins in these 2 methods. The first thing you would need to do is to separate the protein into amino acids by hydrolysis, and then you can further analyze them. So in both examples, both chromatography and gel electrophoresis, different amino acids will move at different rates because of their size and their R groups, so their properties. And these techniques can tell us what amino acids are found and then the general abundance of them in a protein. So again, if you remember from chromatography, you will measure the relative distance each item moved compared to the solvent front. So you do need to know how to do this for paper 3. So you calculate the RF. So it's literally just the distance moved by the solute divided by the distance moved by the solvent. So you would load your samples here, and then you would dip it into its solvent, and then the solvent will carry the amino acids upwards, and they won't travel as fast as the solvent front because they're going to be hindered by the particles in the chromatography paper. So you'll measure this. This is your Y. So the solvent front, and then each amino acid will move a different level, and so then that would be your X from here to here. So when you're doing this, you need to mark your starting point in some way and your solvent front in some way, so that you can remember where they started and ended, because once the paper dries, you won't be able to see either. And then you can just figure out the ratio of how far it moved compared to others. 2D chromatography is kind of fun because it can actually help you to separate all 20 of the amino acids from one another. So you actually will put your sample in 2 solvents. So you'll put it into solvent A, let it run, so you can see that they've all transferred this way. And then you rotate it 90 degrees and then run it again. So then now you have all these particles separating. And so in this example, you actually had a couple of things. So you can see how they've moved in different ways. So that will help us understand better some of the properties and the identities of some of the amino acids. So in this example from our textbook, there were 2 different solvents that were used. So butanol ethanoic acid was the first one, and then phenol was the other one. So you actually had to add a locating agent, which is also known as a dye, to identify where the amino acids were, because they don't have specific colors. So the dye that was used was ninhydrin. All of the amino acids will stain purple in the presence of ninhydrin, except for proline. So the yellow dot you'll know is proline because of its cyclic R group. And then you can identify the different amino acids because they will always move in the same pattern. So you can actually find out what amino acids are there based on former runs. So if you have cysteine will show up kind of here, glutamic acid, and so on. This one only has a trace amount because it's a smaller dot. There's more glutamic acid, and so on. The second technique that we can use is called gel electrophoresis. So in this example, we're using electric current to separate a mixture of proteins. So you can see there's your mix of proteins. You run the current, and then you can see that the proteins have separated. They travel through a porous gel. You apply the current, and then they move at different rates based on their size and properties. Then again, same thing, we have to add a stain to be able to detect the amino acids. So there's two different types of gel electrophoresis. There's native and denaturing. So in native electrophoresis, the proteins are loaded in the gel unaltered, so they haven't been affected, and they are simply separated by their size and shape. In denaturing electrophoresis, they're denatured first, then they're loaded into the gel, and this time they're only being separated by their size. So this is an example from our textbook again. So this is an example of the results from a gel. So in the first laying, a bunch of protein markers were loaded so that we can see how far they move. These numbers here are their molar masses or molecular weights measured in kilodaltons. So you can identify the different amino acids. So in lane B, we can see that there were two lines. So there were two polypeptides. One of them was about 65 kilodaltons. The other one was around 28. C only had one, but it was a thicker line at 28 kilodaltons, and then the last one had one around 32 kilodaltons. We can also run something known as a protein assay, and that's measuring the rate of enzyme activity in the lab. So because the enzyme doesn't get used up, you have to measure either the change of substrate or the change of products. You can't alter or measure the change in enzyme because it doesn't change. So you can do a variety of methods to figure out the enzyme activity. You can either measure the change in the pH over time, the change in color, the change in conductivity. You could measure whether a gas was produced or so on. So there's lots of different ways of measuring the change of enzyme activity. One technique that you can do to measure for proteins is something known as the burette test. So burette, the reagent, is a mix of sodium hydroxide, copper sulfate, and potassium sodium tartrate. If it's in the presence of protein peptide bonds, it will turn purple. So you can use it both qualitatively or quantitatively. We'll talk about how you can use it quantitatively in a moment. So in this example, this was just our burette test, and then one sample had no protein and the other one did have protein. So it actually turns a nice violet or purple color. So we can use a colorimeter to quantify how much protein is in the sample. So light of a certain wavelength will be passed through a cuvette that contains the burette reagent, and the amount of light that's absorbed by the solution will be determined. So you send in light of a certain wavelength, goes through the solution, and then you can measure how much was absorbed as it went through the solution. We can also use something called UV-vis spectroscopy. So it scans in the visible and the UV range and will detect which wavelength is the best absorbed by the sample, and that's known as lambda max. It will be compared against a blank, so the sample without the reagent, or with the reagent but not the protein. So this is the complicated little diagram. So you can go through, you find out, and then it'll detect. So in our first course of biology, we talked a little bit about Beer-Lambert's law. So this law says that the absorbance and the concentration have a directly proportional relationship. So as the concentration increases, the absorbance of light increases in a linear fashion. But this will only work with lower concentrations. The relationship will deviate at higher concentrations, so it won't be linear anymore. And we can actually use something known as a calibration curve, where we look at known concentrations, and then we use that to determine the concentration of an unknown sample. So when we did the concentration of copper sulfate solution last year, when we did successive dilutions, and then we plotted the conductivity versus the concentration, we had a similar relationship. You can do the same thing with this absorbance. We could have plotted the concentration versus the absorbance, and we would have seen a nice linear relationship. The R curve, when we did the conductivity, was actually a little bit more curved.